The cell is the most unusual material that you could find. As a cell is moving around, unlike a car, say, where you have an engine, you have the scaffolding, you have a clutch, the cell basically assembles and disassembles its clutch, its engine, its whole structure, and its wheels even. And so we want to understand how those materials in the cell are assembling and disassembling. And so we want to understand both how these machines work in a healthy, untransformed cytoskeleton, but also how, issues in, say, cardiac disease or cancer metastasis occur because of misregulation of these physical behaviors. A lot of advances in biomedical research recently has been identifying diseases at the molecular or genetic level. So understanding a single gene or how misregulation of expression or activity at a single protein level will result or is correlated to disease of the whole organism. So it will give us a new handle in which to tune and to target therapies to treat human diseases. And the reason this approach is different than what people have done before is there's been a huge advance in our ability to identify the molecular basis of cell biology and understand the structure and function and biochemical and biophysical properties of individual molecules. There are these different proteins that assemble actin filaments into different structures, into different bundles and networks. And it's the mechanical rigidity of these actin networks that enable the cells to transmit forces across its whole body and enable it to change shape and transmit mechanical forces from the molecular up to the cellular level and enable, when a force is applied to the cell, enable it not to be destroyed. We're lying between basic cell biology and condensed matter physics at this point. So I was studying the polymer physics of these materials. Obviously, the next step is to try to understand how these materials are behaving in a living, dynamic, moving, dividing cell. It's a technique where you use high-resolution fluorescence light microscopy, and you inject fluorescent protein into a live cell. You actually use a micropipette to inject fluorescent protein into the cells, and they serve as little light bulbs in the cell. And so you can excite them with a certain wavelength of light and image them at a longer wavelength of light, and take movies, time-lapse movies of these proteins moving around inside a cell as it's migrating, dividing, generating force. To the Pioneer Award, the money really, well, first of all, it's a dream for somebody starting their lab, because in terms of hiring personnel, you know that you're going to have money available for them to just be creative and do their research for the next 3 years when they're a postdoc or as a graduate student, and to really be able to hire personnel with diverse skill sets. So someone with an expertise in biochemistry or cell biology, and combine those with people with a background in condensed matter physics. The expertise in the lab lies in integrating biophysical measurements of forces that cells generate, or applying forces to cells with high-resolution images of the dynamics of proteins. So it allows us to build, say, magnetic tweezers and combine that with imaging. So it allows us to do techniques that I didn't think we'd be able to have the capability or feasibility of doing until 5 years from now or so. So it's very exciting.